Zooxanthellae are single-celled protozoans, dinoflagellates, that are able to live in symbiosis with certain marine invertebrates. In freshwater habitats, similar entosymbionts are known as Zooxanthellae. Most known Zooxanthellae are in the genus Symbiodinium, but some are known from the genus Amphidinium, and other taxa, as yet unidentified, may have similar entosymbiont affinities. Dinoflagellates are microscopic single-celled planktonic organisms that used to be classified as algae but are now considered to be in a large and diverse group known as Chromalveolata. They can be free-living or can be present in large number in the tissues of a host. The motile, mastigot stage in the water column has two flagella and a characteristic method of swimming, while inside a host it metamorphoses into a spherical, non-motile, coccoid stage without any flagella. Zooxanthellae are photosynthetic organisms, which contain chlorophyll A and chlorophyll C, as well as the dinoflagellate pigments periodinin and diadenixanthin. These provide yellowish and brownish colors typical of many of the host species. During the day, they provide their host with the organic carbon products of photosynthesis, sometimes providing up to 90% of their host's energy needs for metabolism, growth and reproduction. In return, they receive nutrients, carbon dioxide, and an elevated position with access to sunshine. Zooxanthellae are particularly associated with reef-building corals, but they also inhabit other invertebrates and protists. Their hosts include many sea anemones, jellyfish, nudibranchs, certain bivalve mollusks like the giant clam Tridacna, sponges and flatworms, as well as some species of radiolarians and foraminiferans. Many different species of Zooxanthellae are present in host organisms, each species with its own adaptive capabilities and degree of tolerance of varying environmental factors. A juvenile organism or newly established colony can acquire its Zooxanthellae via sexual reproduction or directly from the environment. The egg from which the individual developed may have already been infected by Zooxanthellae at the time of fertilization, or cells of the symbiont may have been transferred from the mother in a period during which the larva was brooded by its parent. Alternatively, the new individual may acquire the Zooxanthellae direct from seawater in which the dinoflagellates freely live at some stages of their life cycle. Some stony corals use chemotoxis, with infection occurring as a result of the emission by the coral of a chemical attractant. Infection may also occur after ingestion of infected fecal matter by the host, or of prey that already houses the symbionts. Such indirect acquisition can result in the new host being infected by a species of Zooxanthellae different from that present in its parent, and a bleached coral that has expelled its Zooxanthellae may be recolonized by another species perhaps more suited to the stressors present in the environment.